Hey, thank you, Erica. I'm Pete Hansen, and it's my great pleasure on behalf of the Society for the Study of Reproduction and on behalf of my co-organizer, Dr. Dan Bernard of McGill University, to welcome you to the first of 4 webinars in a series entitled Insights in Neuroendocrinology Reproduction. I don't think I have to tell anybody here that, but I'm going to anyway, that specific neural connections within the brain and neuroendocrine communication between the brain and the rest of the body, particularly through the pituitary, is essential for reproductive processes. It's no wonder then that the neuroendocrinology of reproduction is such an important topic for the Society of the Study of Reproduction. And it's really this idea that animated the organization of this webinar. SSR webinars are a product of the Virtual Education Committee of SSR, and I'm very grateful to the chair of that committee, Joni Jorgensen of the University of Wisconsin, and to Erica Salisbury of our business office for helping to organize the event. I also want to mention the invaluable contributions of Dr. Bernard in identifying the speakers for the webinar series. I also thank all the other moderators of the webinar series, and if you attend all 4, you'll see them in person or through Zoom. Dr. Ian Clark of the University of Melbourne, Terry Nett at Colorado State University, and Dan Bernard himself for their participation. I also thank all the speakers. You really make this webinar what it is. I know you're all very busy, and I appreciate the time that each one of you have committed to bringing us the best research in the field today. You're going to hear more about each of the speakers over the next 4 weeks, but at this time, it's my pleasure to introduce today's speaker, Dr. Sue Mentor. Dr. Mentor is the Fred J. Karsh Collegiate Professor of Molecular and Integrative Physiology at the University of Michigan. Her research focuses on the neuroendocrine regulation and reproduction, specifically the characterization of GnRH and kisspeptin neurons in 4 thematic areas: development and generation of episodic GnRH release, central mechanisms of negative and positive feedback regulation by estradiol, the impact of stress on fertility, and models of infertility associated with hyperandrogenemia, such as polycystic ovarian syndrome. Dr. Mentor is a graduate of the University of Illinois, received her PhD from University of Michigan. She performed postdoctoral research at the University of California, San Francisco. She began her academic career at the University of Virginia before moving back to Michigan in 2010. She's been recognized and honored many times for her research and for her mentoring of graduate students. She's a fellow of the AAAS. And this year, she's being recognized as the Schmidt-Nielsen Distinguished Mentor and Scientist Award from the American Physiological Society. So thanks very much, Sue, and I'm going to just turn it over to you.
Thank you, Pete, and also thanks to the SSR and Joni and Erica and Dan for organizing this series. I think it's going to be an exciting series of talks, and I hope that while it's always exciting to share what our trainees are doing, I hope that we can convince you that the brain really is a major reproductive organ. So what we're looking at here is a sagittal section through a mouse brain. The nose would be on the left, and many factors affect reproduction through the brain. These include environmental signals such as season and time of day, stress, development, and energy balance. And all of these make use of the computational power of the brain to regulate the secretion of gonadotropin-releasing hormone, or GnRH. Now, over the course of the series, my co-speakers are going to talk about many of these areas. Next week, Richard Piet is going to tell you about the diurnal influences on this system. And then in a few weeks, Victor Navarro is going to be talking about energy balance and its interactions in bidirectional manner with the reproductive neuroendocrine system. And then Rodolfo is going to clean up in the beginning of May to talk about how energy balance and development interact to regulate this system. So what I'm going to do today is focus more on the basics, give you a basic introduction to reproductive neuroendocrinology to get everybody onto the same page, and then talk a little bit about our recently published and unpublished work that is going on. So as you're all probably aware, GnRH acts on the anterior pituitary. It is secreted in the median eminence of the hypothalamus near portal vessels that transport the GnRH to the pituitary through a specialized vascular system. In the pituitary, it stimulates the synthesis and secretion of the gonadotropins, LH and FSH, which act on the gonads in both sexes to activate the gonadal functions of gametogenesis and steroidogenesis. And then steroid hormones in particular feedback both at the pituitary to affect the response to GnRH and in the brain to affect the release of GnRH. So as many of you are aware, a major feature of this system is that the release of GnRH is pulsatile or episodic. And these are data from female sheep. They became a very popular model because in order to measure GnRH, you actually have to access those pituitary portal vessels to assay this hormone. You can take LH samples from the jugular blood, but what this shows in an ovaryectomized female over a 5-hour period is a strictly episodic pattern of GnRH, and that each of these GnRH pulses drives a pulse of LH, which allows us in most circumstances to use LH pulses as a pretty good bioassay for what's happening in terms of GnRH release. So these pulses aren't just pretty to look at least I think they are. They have a real function in the control of the reproductive system. So these is a classic ablation replacement study done in ovaryectomized female monkeys by Ernie Nobile's group. And he ablated GnRH neurons by lesioning the hypothalamus, and then he replaced GnRH in different patterns. So for this first segment, he gave GnRH at 1 pulse per hour. And what we're looking at is daily now measurements of luteinizing hormone and follicle stimulating hormone to see if these replacement regimens that he gave to the monkeys restored gonadotropins to levels compatible with fertility. And when he gave 1 pulse per hour back to the monkeys, LH and FSH levels were restored. But when GnRH was restored in a continuous manner, it turned out that constant GnRH is a real turnoff at the pituitary, and LH and FSH levels were downregulated in a reversible manner because restoring pulsatile replacement was able to bring back the gonadotropin levels. So this observation that continuous GnRH suppresses pituitary output is still used clinically today, for example, in treating precocious puberty. So this was a 1 pulse per hour treatment, and the data I showed you on the prior slide also indicated a frequency of about 1 pulse per hour, but I don't want you to leave here with the impression that GnRH is always released in an hourly manner. Actually, the ability to modulate the frequency of GnRH release is key to its function during the cycle. And in another experiment in the same model system, Nobile's group demonstrated this by changing the frequency this time from 1 pulse per hour, lowering the frequency to 1 pulse every 3 hours. And what you can see is that these lower frequencies favor an increase in FSH and a relative decrease in LH, which again is reversible when you change the pulse frequency back. And these frequencies represent very closely what is observed during the normal female reproductive cycle. Here I've illustrated it as a human menstrual cycle, and my hypothalamic output is quite schematic. But as the frequency of GnRH changes through the follicular phase, this helps drive the differential release of FSH and LH. And this was an important discovery, this pulse frequency dependence, because at that time inhibin was still just a hypothesis, although we know it's important in this now. And it had been a mystery how one neuroendocrine factor, GnRH, could potentially produce these different levels of FSH and LH that we know are necessary for driving good follicular development in the early follicular phase, followed by an acceleration of steroidogenesis to induce the preovulatory surge later in the follicular phase. So my lab is really GnRH neuron centric, but I would be amiss if I did not tell you that GnRH neurons are not in this alone. First, as I've already alluded to, GnRH pulse frequency is sculpted by gonadal steroids, but the GnRH neurons themselves do not express detectable levels of most steroid receptors. So here on the upper right, you can see a dual immunohistochemistry for GnRH neurons shown in brown and estrogen receptor alpha in black, and there's no overlap. And back in the time before free color figures, yes, we used to publish multiple colored figures in black and white to save money. You can see a GnRH neuron with a clear nucleus and nuclear progesterone receptor staining. So not much indication of steroid receptor expression in these cells, despite the responsiveness to it. And so this really led to a longstanding hypothesis in the field that steroid feedback is due to upstream elements. And most people have thought that these are neural, and indeed the favorite neuropeptide in the field right now is probably kisspeptin. These are two papers that were published in 2003 showing that what was then called GPR54 and is now called the kisspeptin receptor, when there are mutations in this receptor, patients have hypogonadotropic hypogonadism. And this is a receptor for a neuropeptide called kisspeptin. So in my lab, the way we study this system is to use transgenic reporter mice to identify neurons. We make brain slices from these animals and we use them the same day. So this is not a long-term culture. And we then use that reporter to identify the cell of interest. Here is a fluorescent GnRH neuron, and here is a recording pipette attached to that neuron in the milieu of the brain slice. So one of the methods that we can use is to very lightly attach this pipette to the GnRH neuron and record extracellularly the currents that are associated with action potential firing. And when you do this under control and kisspeptin situation, what you can see is there's firing under basal conditions, but when you treat that brain slice with kisspeptin, the firing goes up enormously. So this is really the strongest activator of GnRH neuron activity that has been studied to date. And there was even more increased excitement about kisspeptin when the cells that make it were localized, because these are in the arcuate nucleus of the hypothalamus, and the anteroventral periventricular area, which I'm going to call the AVPV. And these areas had long been associated with neuroendocrine control from old lesion type studies in which they disrupted different parts of reproductive function by lesioning these two brain regions. And more to be the point in terms of steroid feedback, both of these populations express multiple steroid receptors. It's very interesting how the expression of kisspeptin is regulated by estradiol feedback. It is very different in these two cell types. So the arcuate kisspeptin neurons, which I'm going to call candy neurons on and off, that's what the field calls them because of the other neuropeptides expressed in them. In an intact female, there's low levels of kisspeptin expression. These are data from Jeremy Smith and Robert Steiner's group. When you take out the ovaries and remove negative feedback, kisspeptin expression goes up. And when you replace negative feedback, it's restored back down. In very marked contrast, in the AVPV, kisspeptin expression is high in the intact animal and goes down when you take out the gonads and is restored by estradiol feedback. So the regulation is quite the opposite, and that has led to sort of a current dogma in the field that these two hypothalamic kisspeptin populations control these two major modes of GnRH release. Specifically, the arcuate nucleus is in charge of pulsatility release, and the AVPV is regulating positive feedback and the GnRH surge. But when you've got the brain, you've always got to be asking the question, is there really just one ring? And I would argue probably not. This is a book that was published by Andrea Gore the year before kisspeptin was discovered as a neuroendocrine regulator of reproduction. And it has over 300 pages of other neural systems that likely regulate the output of GnRH neurons. And nowhere in that 300 pages does it even mention half of the cells of the brain. There's not even the word glia in the index. So what are glia? These are non-neural cells in the brain that are absolutely vital to central neural function. And they come in three main flavors. There's microglia, and these have a lot of immune functions. There's oligodendrocytes, and these support axons through myelination. And then there's the astrocyte, which is what we're going to spend most of our time talking about today. Although I will take a short detour into a completely unrelated project that I think is pretty cool and is getting close to publication. I want to show you what one of the other postdocs in the lab is doing. So one cannot even pretend to be a neuroscientist if you don't occasionally show one of the beautiful drawings from Santiago Ramon y Cajal. And these are his drawings of astrocytes in the human hippocampus. And there are these darker cells that you can see here. And he really, as he did with many of his drawings, captured several essential features of these cells. First and foremost, their structure. They have a somatic area and then lots of different processes radiating out like a star. And this is where the term astrocyte actually came from. But also that they're very compartmentalized, having these, not only these processes versus a somatic area, but they sort of tile themselves so that they don't overlap a lot. They interact at the edges of their domains, but they sort of fill their own three-dimensional space. They also interact very intimately, as you can see in this case, with the neurons. There are as many astrocytes in the brain as neurons, and for a long time, they were thought to just be structural cells. In fact, the term glia comes from the Greek word for glue, but now we know that they are very vital in terms of maintaining energy homeostasis in the brain, in synapse formation and function, and also in bidirectional communication with neurons. And in this regard, astrocytes are working components of what's become known as the tripartite synapse. So in most neural communication, there's a presynaptic neuron that will release a transmitter through a gap called the synapse to activate or suppress a postsynaptic neuron and change its neural activity and its neural secretion. The third component of this tripartite synapse is the astrocyte, and it can modulate synaptic efficacy in a couple of different ways. One way it does this is to take up neurotransmitter into the astrocyte, so to remove it from the synapse and stop the signaling. Another way it can do that is to send its processes to actually interfere physically with the synapse. And these are really plastic cells. And one of the things that we know regulates astrocyte plasticity is steroid hormones. In addition to modulating synaptic efficacy, these astrocytes can respond to neural and other signals. So they have receptors for neural signals, and they can also secrete their own substances that can act back on the neurons. And this is called gliotransmission. And it was really in vogue for a while, and then it went out of vogue and became controversial. And now the pendulum seems to be swinging back towards a happy medium in which gliotransmission is likely to occur, but it doesn't look exactly like neurotransmission, and more work needs to be done. So Charlotte Vanacker was a postdoc in the lab, and she was very interested in GnRH neuron glial interactions. And so she took this picture of a green GnRH neuron surrounded by GFAP, or glial fibrillary acidic protein expressing cells. And this is an intermediate filament protein that's prominent in astroglia. And the questions that Charlotte was really interested in asking was whether astrocytes are involved in GnRH and LH release through communication with GnRH and or kisspeptin neurons, especially those in the arcuate nucleus, in order to change the pulsatil output of GnRH. And the approach she used was something called DREADs. These are designer receptors exclusively activated by designer drugs. And these are much more used within the neuroscience community than outside in other parts of the body. So I'll just go over a little bit about what they are. So these are modified G protein coupled receptors, modified from the acetylcholine receptor. And you can use a synthetic ligand such as CNO or clozapine and oxide to activate these receptors. So when you put CNO into the bath, it's going to bind to that DREAD and activate the signaling pathway to which that DREAD is coupled. And we specifically use GQ coupled DREADs, and that's going to increase IP3 and intracellular calcium. Now, acetylcholine no longer binds to these receptors, so we don't have to worry about it. So we have a way of genetically targeting the astrocyte by putting this expression of these substances under control of that GFAP promoter and putting it as the payload in an adenovirus vector. And we also can include M-cherry, which is a fluorescent marker, so we can look at where the infection took place. And M-cherry in addition to the dread. Now, to get spatial resolution, we can target stereotaxic injections to different regions of the brain. So we did the preoptic area to look at GnRH neurons, excuse me, and the arcuate nucleus to look at the candy neurons. So first we have to do some controls, specifically looking at the effects of CNO, especially when there's actually a paper out there that suggests that the drug that you're using might possibly be evil. And they have a good point. CNO is metabolized relatively quickly to clozapine, which is a neuroactive substance that is still used as a treatment of last resort for schizophrenia. So the idea that it might be affecting neural function is something we definitely have to think about. Luckily for us, a former student in the lab, Eden Dulka, had done these controls for her experiment. And this is a pulsatil LH release pattern from an overreactomized mouse. She treated with saline, saw no effect on pulses. She treated with CNO, and it looked just like saline. So no effect of CNO on pulsatil LH release. She also did a couple controls with clozapine, looking at LH release in vivo, and there was no effect over a 30-minute period. And there was no effect of firing rate on a couple of GnRH neurons. So we feel reasonably confident that the effects of CNO are going through the dread receptor rather than it's an off-target effect on other neurons. So Charlotte stereotaxically targeted these AAV viruses with either the mCherry control or both the dread and the mCherry to the preoptic area. And here's immunofluorescence showing the area that was infected, and these blue areas are the branches of the anterior commissure for the aficionados out there. If you blow this up, you can see that this is surrounding the GnRH GFP neurons. And then for the candy neurons, she used TAC2 GFP males, and TAC2 is the gene that encodes for neurokinin B, and it's expressed in the kisspeptin neurons, and you can see these neurons are in the infected area. So this approach of using GFP was pretty effective at targeting mCherry expression to astrocytes. And here we detected the astrocytes by S100 beta staining, which is another astrocyte protein. And if we look at the bottom, you can see about 91% co-localization of the mCherry signal with the S100 beta. There are a few neurons, however, that express the neural marker NuN and also the mCherry. About 7% of the cells infected with mCherry are neurons. And this is not really surprising because these cells come from a common progenitor, and it's entirely possible the GFAP promoter doesn't get completely turned off. But it's something we need to think about when we interpret our data, that there are a few neurons that are going to be expressing this. So the first question Charlotte asked was whether expressing GQ signaling in GFAP expressing cells would alter LH release in vivo. So to do this, she acclimated mice to the sampling procedure for about 3 weeks. And then she took tail blood samples at 10-minute intervals and injected cells with the animals with vehicle and then CNO. And when she looked at the LH patterns, what you're looking at is LH over the time. These are the individual LH secretory profiles. The pink line shows an animal in which we did detect neurons with cells with a neural morphology that contained mCherry, suggesting there might be neurons infected. So we're showing these data here for transparency, but we did not include those in our statistical analyses. So when there was just mCherry targeted to the GFAP expressing cells, there was no effect of CNO on LH. In contrast, there was a really large increase in LH release when the GQ receptor was targeted to the GFAP expressing astrocytes. So CNO markedly increased LH release. When the same experiment was done in the arcuate nucleus, and here we have a little bit more complication because we had to do this bilaterally instead of just down the center of the brain. So the dotted lines show unilateral hits and the solid lines show bilateral hits. The pink again shows cells with unidentified neurons infected with the dread. And the blue lines show actual TAC2 expressing neurons that were infected with mCherry. But again, just mCherry, no effect on LH. But unlike GnRH neuron area, when you activate GQ signaling in the arcuate region, the LH release does not go up in a consistent manner. There's 2 cells that go up, in which there was neurons infected in that animal, and there's 1 animal that goes up with in which the preparation appeared clean. But the vast majority of these are not responding. And if we look at the summary data, you can see the clear increase in LH when glia in the area of GnRH neurons were activated to have GQ signaling, but not in the area of the kisspeptin neuron. So this looks like it might be a kisspeptin independent way to control GnRH, potentially GnRH release leading to LH release. So Charlotte then moved into brain slices so she could start to approach mechanisms. So we made the brain slices as before, and she did those targeted extracellular recordings to look at the currents of action potential firing. This shows the timeline of her experiment. She did a control period, applied CNO for 6 minutes, and then washed it out. So these first data are from the pre-optic area. And let me orient you to the graphs because there's going to be several that look like this. So on the top, we have a representative example of the firing rate over time in the experiment of a neuron, a GnRH neuron. And this is a pre-optic area with just the M cherry. These little letters A, B, C show where these swaths of raw data are from. So you can see what the actual raw data look like. And on the bottom are the individual traces showing the change in firing rate over time for the CNO period and the washout period. So when M cherry is targeted to, with M cherry to the GFAP alone, no change in firing rate of GnRH neurons. But when we include that GQ dread and target that to the astrocytes, we see a marked increase in the firing rate of GnRH neurons. Now, it might seem kind of crazy, so I should point out, these were all GnRH neurons within the viral hit. And it might seem crazy that we'd even look outside the hit, but one of the reasons I really got interested in glia as a mechanism in reproductive neuroendocrine function is that tiling structure that I told you about before, because glia can communicate over relatively long distances through calcium waves. And we're still looking for mechanisms for which GnRH neurons can be coordinated to produce pulsatil release. And so Charlotte recorded from GnRH neurons outside the viral hit. And unfortunately, we didn't see any evidence that activating glia in another part of the brain slice could actually change the firing rate of GnRH neurons. So that was kind of a sad day when you saw those data, but the data are the data. So what does it look like in the arcuate nucleus if we do this experiment? Well, it sort of looks like the LH data. There's no effect in the controls of CNO. There is no effect in the animals that have GQ targeted to the astroglia in the arcuate region of applying CNO. And I should mention that CNO is shown in this yellow shaded area. And this is whether we are looking in a hit or a miss. So the individual data are shown here. And similar to the LH data, it doesn't look like activating glia in the area of the arcuate nucleus is leading to a change in candy neuron firing rate. So here's that detour I promised you. And now for something completely different, as John Cleese would say. I want to talk a little bit about the relationship between firing rate, since we're using that as a readout, and the actual secretion from these cells. And to approach that, we've used a method called fast scan cyclic photometry, in which you can do an oxidation reduction reaction on a carbon fiber microelectrode. Now, the structure of GnRH is here, and I've circled 2 oxidizable amino acids that it has. And Kasia Glanowska is a former student in the lab, and she worked out a voltage protocol to apply to those carbon fiber microelectrodes that produced a current that was specific to GnRH. And so what you're looking at here are heat maps of when that current appeared. On the left is GnRH release evoked by high potassium. You can see this nice green band showing high current, suggesting high GnRH release. The middle here shows spontaneous GnRH release detected in the median eminence. And this right panel is a control done in a hypogonadal mouse, which is a spontaneous GnRH knockout. And you see absolutely no GnRH signal in these animals, even when you evoke with potassium, showing that this other signal is specific to GnRH neurons. So C. Chen, who's a postdoc in the lab now, is doing this really terrifically complicated experiment to ask a very basic question, which is, does firing rate of GnRH neurons relate to the amount of hormone that's secreted? And so what she's done is scan brain slices for a situation like this, where she can see two GFP identified GnRH neurons, and then she puts that carbon fiber marker electrode right where those cells appear to be interacting with one another. Of course, we can't tell if they're interacting at this light level, but that's where she puts the electrode. The reaction to produce that current has to take place on the electrode. So this is a really local measurement of GnRH release. She then took a recording pipette and made a whole cell recording of one of these cells. And the type of recording she did was a voltage clamp recording, so she could manipulate this membrane potential of this cell and make it look like it was firing. And to do this, she used the most boring GnRH neuron action potential we could find. It had the median characteristics of everything. All its parameters were within the 95% confidence interval. And its pre-firing period, so this part here, was the duration of the interspike interval during a burst of firing. It was very close to both the mean and the median. So when she strung that template together into 2, 5, and 15 action potential trains, she could ask the questions of whether a change in firing causes a change in GnRH release, and whether stimulus secretion coupling is affected by steroid feedback. And she could ask this because she did this both in sham males and in orchidectomized males. And so what she found is that in the sham operated males, as you increase the number of action potentials applied to that GnRH neuron, the amount of GnRH released at that junction between those two cells did increase. And what I think is super interesting, and an area that I think a lot of people can exploit for future research, is that in the gonadectomized animals, the stimulus secretion coupling was actually much more sensitive. So the increase in GnRH occurred with far fewer action potentials, suggesting that the stimulus secretion coupling is another mechanism to target for understanding how opening the feedback loop increases the release of GnRH, and possibly how other things such as positive feedback increase the release of GnRH. So that's a little detour I wanted to send you on. And I think it provides us some context for looking at the summary data for the last two glia-related experiments I showed you, because we can now say that this increase in firing rate of GnRH neurons in the preoptic area, when those cells, those glia were infected with the GQ dread, this is likely related to an increase in GnRH output. And again, in the arcuate region, we saw no consistent change in the firing rate of these cells. So this brings up two interesting questions. So how do astrocytes communicate with GnRH neurons? And are candy neurons devoid of that mechanism? And is that why they don't respond? So one of the likely suspects is prostaglandin E2, or PGE2. It's been known for about 50 years that if you inject this into the brain, it will increase LH release in a dose-dependent manner. And more recently, Vincent Prévost's lab showed that PGE2 can increase the firing rate of GnRH neurons in brain slices. So the reason PGE2 is interesting is because astrocytes express the rate-limiting enzyme for synthesis of prostaglandin. It is thought to be one of the gliotransmitters, and GnRH neurons express both EP1 and EP2 prostaglandin E2 receptors. And so what Charlotte did was to first do some control experiments. She looked at the firing rate of GnRH neurons in response to dimethyl PGE2. Now, she used dimethyl because PGE2 is really labile and we wanted to use a stable form. And she saw the same sorts of increase in firing as other people had for GnRH neurons. She then identified a dose of a cocktail of EP1 and EP2 receptor antagonists that could block this increase in firing. And so you can see the summary data here. And the reason she did that is she wanted to ask the question of whether blocking EP1 and EP2 receptors would block the effect of CNO activation of that dread receptor on the firing rate of GnRH neurons. And it was pretty effective. On the left here, you see an example of a cell that did not respond to CNO with an increase in firing rate after blocking EP1 and EP2 receptors. And in the middle, a cell that had a slight and delayed increase in firing rate. So blocking these receptors reduces the effect of activating glial GQ signaling on GnRH neuron firing rate. So what about the candy neurons? Well, prostaglandin E2 didn't affect the firing rate of candy neurons. This shows actually 9 recordings. They're so similar, they're actually overlapping. And just to show you the comparison in the GnRH neurons, that the PGE2 was very effective at increasing firing rate. So that suggests that the astrocytes can activate the GnRH neurons through a mechanism that doesn't seem to affect the candy neurons, or doesn't seem to be a response to these prostaglandins here, in contrast to the GnRH neuron, and that this is, again, a potentially kisspeptin independent way of regulating the output of the reproductive neuroendocrine system. So lots of problems with these studies. First of all, we only did males. Secondly, we only looked in the morning, and this is a notoriously diurnal system. And finally, we're using this CNO stuff. We have no idea what the endogenous activator of GQ signaling is in these cells. So this is where Christian Phillips took over this project as a new graduate student last fall. And he wants to ask the questions of whether the efficacy of CNO and DREAD signaling varies with sex, time of day, and cycle stage as his first project. Now, he is looking at diestrus versus proestrus in the females, but these are still pretty early days for the proestrus. And so we're going to look at comparisons between the males and the females in diestrus at 2 different times a day. And the diestrus female is a negative feedback condition like the male, thus is probably a best place to start with comparisons. So what Christian has done is to inject 2 different viruses at the same time. So he used GFAP to drive expression of a genetically encoded calcium indicator, GCAMP6F, as well as either the GFAP mCherry or the GFAP DREAD that we've used in the prior studies. And again, he injected into the preoptic area or arcuate, but in all these different groups. And we know that the GCAMP6 is effectively targeted to S100 beta expressing neurons. So again, good viral targeting with this AAV construct. So what Christian did was to do calcium imaging and identify a number of regions of interest that had the fluorescent signal. And he plotted these as relative plots. So we're looking at the calcium within a cell over time. And these are cells that are mCherry controls, and applying CNO does not change their calcium profile. And each of these is an individual cell. So you can see quite a number of cells within a brain slice. When he looked in the cells that had been, where the glia had been infected with the DREAD, there's a really sharp increase in intracellular calcium in all of these astrocytes that were infected with the DREAD. And so when he looks across the various groups, you can see that the controls are these dotted lines at the bottom, no intracellular calcium increase in those, but robust increases in intracellular calcium in both males and females in both brain regions, preoptic area on the left and arcuate on the right, and at both times of day. So the lack of effect in the prior studies of the arcuate in the arcuate nucleus were not because the CNO was failing to increase intracellular calcium in the DREADs. We are seeing an emerging sex difference that's possible in the afternoon in the arcuate nucleus, and we'll have to see if that holds out as we gather more N. So Christian is also starting to look at whether endogenous activators of intracellular calcium can increase endogenous activators of metabotropic receptors, I should say, can increase intracellular calcium in hypothalamic glia. So in this model that I've been showing you, I've been having this little black receptor to show the DREAD. What we really want to know is what this blue receptor is, which is what would be endogenously activated to induce the signaling pathway. And so Christian is looking at astrocytes within the preoptic area, and he wants to apply agonists of different candidate receptors, but we need to remember that when we back apply something, we're actually hitting the whole slice. And so to limit communication within that slice, he blocked receptors for both ionotropic GABA and glutamate fast synaptic transmission, which is a major mode of communication in the brain. And in some of these early studies, he also blocked action potential generation, which should further reduce neuromodulator release and let us know more with more confidence if the receptor agonist is acting on the astroglia itself. So the candidate he tried was something called ACPD, which is an agonist of type 1 and type 2 metabotropic glutamate receptors. And what you can see here are the area under the curve for the calcium imaging, and the red or the black lines show when ionotropic receptors were blocked. And you can see a nice increase in intracellular calcium in these three slices. In blocking tetrodotoxin on top of that, you still see an increase in intracellular calcium in 2 of these 3 preparations. And this is providing some early encouraging data that a type 1, type 2 metabotropic glutamate receptor agonist is a potential, the metabotropic glutamate receptor agonist, metabotropic glutamate receptors are possible endogenous activators of this signal. And that is something that has been seen in other brain regions, in particular mGluR5. So I want to return to the calcium response before I show you the last piece of data. This shows again the AM and PM preoptic area, and I want you to look at the pink line here, which is from the females, and it's really quite similar between these 2 different times a day. So the ability of the DREAD to increase intracellular calcium does not seem to depend on time of day. And that's an important technical control. We want our method to be similar in both of our groups. But we don't know anything about what's happening post-calcium in the signaling pathway and whether that might change with time of day. So these are super early data from Christian's work, but here is the morning samples for LH release in these females. And just like we saw on the males sampled in the morning, CNO is inducing an increase in LH release. This is not happening in the 2 animals, and I emphasize that 2 animals that he's looked at so far on the afternoon of diestrus. So despite that increase in intracellular calcium in the GFAP expressing cells, this does not seem to be translating downstream to an activation of the reproductive neuroendocrine system. So these actually match up with some data that Charlotte had gathered on firing rate, in which she looked at the firing rate of GnRH neurons in the late afternoon in diestrus females and saw no effective CNO. And we were excited by these data, but we were also worried because of this guy. These animals were injected with the virus right before the University of Michigan locked us down long and hard. And our typical survival period is 3 to 5 weeks post-injection, and these animals were around for months. And although these particular slices looked clean in terms of neural infection, we saw a lot of neurons expressing the M-cherry marker, which is very atypical for the preoptic area, a little bit more typical in the arcuate, but we were just a little bit too nervous to go ahead with these data, but they seem to be matching up with what Christian is seeing. So what I hope I was able to convince you of today is that the astrocytes are a likely important aspect of reproductive neuroendocrine function, and that they can activate GnRH neurons likely through a prostaglandin-dependent mechanism that may vary with sex and time of day and steroid feedback condition, and that this seems to be something, at least in the examples we've studied so far, that is independent of these arcuate kisspeptin neurons. Now, well, another thing that I should tell you is that we also have evidence from older studies that GnRH neurons themselves may be functionally glutamatergic, as we've shown in other studies that they can activate what seem to be astroglia to change synaptic transmission back to themselves in a mGluR dependent manner. So there's possibility for a cute little circuit here that is independent from these kisspeptin neurons. Now, even though I don't think there's one ring, I don't want you to get the idea that kisspeptin neurons are not important. Kisspeptin is super important for this system, and I'm sure the other 3 speakers in this series are going to tell you lots of interesting things about kisspeptin. But I think it's better to think about the hypothalamus much more like the interactions you see in Game of Thrones. And yes, I am desperately waiting for the winds of winter, but while I wait, I will thank you for your attention and also thank the people in my lab who collected the data I shared with you today. I'm happy to answer any questions that you have.
Hey, thank you very much. That gives us a lot to think about. And we do have a question and answer button at the bottom of your screen, and I'm going to read the questions off, and Sue is going to give the answers. And there's also a chat button, and I'll look there as well. So I'll just do these one at a time, Sue. I know it's not.
That will make it easier.
I know it's not the usual way. But the first question is from Mike Lehman. Great talk, Sue. I'm sure someone else will ask. Well, not really, because he goes first. But were there any effects of CNO in the female arcuate? Given sex differences in candy cells, who would be interested to know?
On the docket to do. It's a great question. And on that last slide, I didn't point out the funding, but Christian's NRSA, which has that project in it, hopefully got a fundable score. So he's locked and loaded to do those studies.
And then Rodolfo Cardoso asked, astrocytes in the median eminence play a key role in regulating the permeability of the blood-brain barrier. Could astrocytes in the median eminence interact with GnRH neuron dendrons to control GnRH release?
So that's a great point, and it was actually something I meant to mention because the GnRH processes do project right through that infected arcuate region on their way to the median eminence, and we still didn't see that activating GQ signaling in the arcuate nucleus was effective at changing LH release. And so what that might be suggesting is that the effect of the astrocytes is not only region of the brain specific, but region of the GnRH neuron specific in terms of that prostaglandin effect. So I think that that's an interesting question that is going to require further work to answer more fully. But the GnRH, the terminal part of the GnRH axons would be going right through that area.
Thank you. Dan Bernard asked two questions, but I'll take the first one first. Great talk, Sue. In the first in vivo DREAD experiment in astrocytes, can you conclude that the effect on GnRH release and the POA is kiss dependent? This is more than one question, Dan Bernard. Would you see the same result in a GPR54 knockout or in the presence of a KISS1 receptor antagonist?
So I think you were asking if it's kisspeptin independent. So we have not done that, and the antagonist experiment, and we have not done the knockout experiment. I think those would be good to do. But since there is no effect on kisspeptin neurons, we can't conclude it's kisspeptin independent, but it seems to be independent of activating the firing of the kisspeptin neuron. I think we can say that. Maybe it's neurokinin B.
He also asked, how long is LH elevated following CNO treatment? Is it sustained as long as the CNO is present, or is it pulsatile?
So that's a great question, and I can't answer it because of the limitations in blood sampling for the mice. So we can't keep sampling forever. And Charlotte, actually, we didn't know what was going to happen, and so I showed you the first 90 minutes of sampling, and after that, she injected GnRH just to make sure the pituitary could respond. But we were really just looking at the response, and we haven't actually assessed the duration of how long LH stays up. It didn't look like the start of a pulse. It went up and stayed up in that short observation window.
Lots of questions. Mauro Silva asked, he also said, great talk. As you do not see differences in calcium increase between the morning and afternoon in females, can we consider a difference there might be in the morphological interaction between astrocytes and GnRH neurons?
Yeah, I think that's a great point. So the calcium experiments are really technical controls to convince ourselves that it's not something to do with the infection efficacy that's leading to any changes. But I think anything post-calcium signaling is completely open for mechanistic investigation. That includes, the receptors expressed on the cells at these different times, the morphology of the astrocytes themselves, the synaptic plasticity of the GnRH neurons themselves. So all of those are potential things to look for in terms of how this response may vary with time of day, with sex, with region of the brain.
Lori Reitzman asks, do you plan to do any breeding studies in mice with CNO activated GnRH related astrocytes?
We hadn't thought about it. One of Charlotte's other projects that got COVIDed was trying to stop cycles with GI dreads, so inhibitory dreads. And again, that was the experiment we were going to try to do, but we hadn't really done anything with the GQ.
Okay. Alexander Sasha Kaufman says, great talk, Sue. I think this must be a Game of Thrones. Winter is coming. Have you looked at inhibitory dreads and astrocytes and their effects on GnRH or LH?
So Charlotte did a little bit of calcium imaging with that. And unlike other brain regions where GI coupled dreads, and this is up in the hippocampus, GI coupled dreads can actually increase intracellular calcium in the astroglia in the hippocampus. We didn't do a huge N. We did, I think, three or four preps, animals, and we did not see the increase in intracellular calcium. What we need to do is screen GI receptor expression in astroglia in the hypothalamus to see if there's anything there and whether that's worth pursuing. But like I said, the in vivo experiments we had planned kind of got derailed by the pandemic, and we haven't gone back to that yet.
Jordika Koss asked, would glutamate uptake and conversion in astrocytes change during the cycle? In other words, hormone-dependent changes in enzymes.
So their glutamate transporters could change. Glutamate packaging could change. Glutamate receptors could change. All of that could. I wouldn't rule any of that out as a possibility. The GnRH neuron itself doesn't seem to get a lot of glutamate input in terms of ionotropic currents. We think they're in the slides because we can tweak them and make them active, but I'm really curious as to what the metabotropic receptor profile and how that might change, and especially how that might change on the astroglia and also on the presynaptic terminals.
So Dr. Koss followed up, says, is it known that they change, or you're speculating?
So I'm not even speculating. I think it's a reasonable thing to look at. The level of work that has been done hasn't gotten to the single cell level for this. It's sort of like if you punch out this part of the brain, you can see changes in receptor mRNA, but you don't know that mRNA goes to protein. You don't know the protein goes to the same part of the cell or to the same extent. So I think, the studies that we have access to that might indicate that things change in terms of expression really have a lot of steps to go through before we get to see whether there's actually a functional change.
Maybe this is a question I have, and maybe it's not technically feasible because maybe an astrocyte next to a GnRH neuron is different than an astrocyte somewhere else. But would RNA-seq or single-cell RNA-seq to look specifically for receptor genes give you an idea, like is estrogen receptor, progesterone receptor, whatever receptor expressed on those cells?
So one of the things that Christian has proposed to do as part of his dissertation project is, I don't know if we'll go all the way to RNA-seq, but we have used translating ribosome affinity purification, where we label the ribosomes, the L10 ribosome specifically, with GFP and pull down the translating messages. I think that is sort of our plan, to try to screen the astrocytes in the preoptic area versus the arcuate region. And the region we probably won't go to RNA-seq is the GPCRs really require a lot of amplification to see because they're not expressed at super high levels. So I think we'll save the expense we have some nice linear amplification methods we set up in the lab and just use those and screen for our candidates. And steroid receptors would be in there. That whether they've been shown in astrocytes in other parts of the brain, but the data in the hypothalamus really are not super consistent as to whether they're there or not. So I think that's an important question to address. Although we know that steroids can affect cells that don't express the receptors, because look at GnRH neurons.
So we have 6 minutes. I'm going to try to get all these questions, but if not, I'm sure.
Email me.
Yes, very good. Thanks. Dan Bernard asks, are there effects of COX-2 inhibitors on pulsatility LH release?
So there have been studies with indomethacin, and I'm blank. Sergio Ojeda did some of these where he put, I think he put indomethacin in different regions. I know that indomethacin blocks the GnRH mGluR feedback circuit to GABA neurons that impinge on GnRH neurons. So there is some indication of the prostaglandin receptor inhibition, prostaglandin synthesis inhibition affecting the neuroendocrine system.
Dan had one other question.
Dan never has just one other question.
Do you think any steroid effects on GnRH release are mediated via astrocytes in the preoptic area?
I wouldn't rule it out, but we don't know yet. That's sort of where I see this going longer term. But I think we got to nail down our basics first. But I think it's a great question.
Let's see. I think I got all the questions that people have asked. If anyone has another question, we still have a few minutes. That probe that you used to measure GnRH, how do you know that's specific? I know you got those residues on the amino acids at GnRH that react with the probe, but can you be sure there's not others? I know you did that control with the knockout.
Yeah, the control with the knockout is what convinced the analytical chemists. So they're convinced it's something related to GnRH. They're not convinced it's related to GnRH. In other words, it's an oxidative or reduced product of GnRH. So in some substances, when you do cyclic photometry, it's a cycle. So you'll get both the oxidation and the reduction. With peptides, and you can really only do small peptides because the oxidizable amino acids tend to be hydrophobic and they'll fold inside if they've got a chance. And GnRH being only 10 amino acids, they can't hide. But usually with peptides, the reaction, there's secondary reaction products, and so it doesn't come back. And that's what they think. We developed this with Jill Benton, who does FSCB development for a living. And we only see the oxidation, we don't see the reduction. So, but they were convinced by the hypogonadal mouse control, because from what we know of people looking for other neuroendocrine factors in that animal model, they are basically intact. So all the other neuroendocrine things should be secreted. We've also looked independently at a couple likely suspects like dopamine and kisspeptin, and neither of those produce that signal that produce a current with that voltage protocol we're doing. And with the FSCB, you can tailor the shape of that to really get really specific as to the substance that is oxidized. Oh, you're muted, Peter.
Maybe this, because my dog was barking again.
Oh,
maybe.
My dog is probably barking too.
Maybe this will be the last question. Richard P. asked, any plans on looking at, I think you really got our attention on astrocytes. Any plans on looking at astrocyte-mediated regulation of preoptic kisspeptin neurons in females or males?
It is in the plan. In fact, one of the things we wrote into Christian's NRSA is that he would look for an undergrad to mentor on that project. So we are taking applications now. So yeah, I think because we've totally ignored the AVPV kisspeptin neurons in this, and mainly because in the beginning we were just doing the males, but it's definitely a place to look. That's the problem with publishing early unpublished data, is there's more questions than answers. But
I'll take one more question from Ana Clara Santana. Is there any interactions between astrocytes, candy, and theta neurons? Is that how you say that?
Theta.
I knew I'd get it wrong.
Tuberoinfundibular dopaminergic neurons. I don't think anybody's looked. So those cells, the theta neurons and the kisspeptin, arcuate-kisspeptin neurons, are right in the same region. And I'm not aware that anyone has looked.
Do we have a little time, Erica, or is it going to close at 6?
It is completely up to you and Sue. If you're happy to stay and answer a few more questions, you're more than welcome to, but we also could wrap up here. I could even drop contact information into the chat if that would be preferable to you.
I have 2 more questions and we'll end it. So one is from Dan Bernard. Have you tried the DREAD experiments and the AVPV? Any effects on surge release?
Haven't done it yet.
And then Renata Frazao says, I believe my question is in line with one of Dan's, but she's curious to know if the virus injections in the preoptic area exclude the AVPV. Or could this nucleus have been contaminated?
That's definitely a possible, especially the more rostral parts. So we were trying to get good GnRH neuron coverage, and there is some overlap in those populations. So I would not exclude that possibility.
Well, I want to thank you very much for
my pleasure. I'm really glad SSR is doing this.
Exciting seminar and a great way to kick off our webinar series. And I thank everybody who participated. Really great questions. Thanks very much for everybody's participation. Thank you, Erica. And we'll do this again next week. Thanks a lot.